Thank you for attending the latest installment in the educational series presented by Princeton Applied Research. Today's topic is the Scanning Electrochemical Microscope, SECM. The VersaScan is our scanning electrochemical workstation. So pictured here is the VS base. This includes the three-dimensional positioning system along with a vibration-resistant stainless steel base with honeycomb design to minimize the transfer of vibrations from the laboratory environment into the positioning system. Pictured here is also both cells, the VSL cell, which is a 1 liter cell, and the VSML cell, a 7 milliliter cell. This VS base can be packaged with many different types of electrochemical measurements to do localized electrochemistry. Here pictured is the setup for a scanning electrochemical microscopy, SECM. We have pictured here the two Princeton Applied Research potentiostats, and interface to the front of the VS base is our SECM probe. The goal of the scanning electrochemical workstation is to provide spatial resolution to different electrochemical and materials-based measurements. And so we'll discuss a little bit about that and a little bit of the key applications and the fundamentals of this technique. A key component of a scanning electrochemical workstation is the positioning system itself. There are two ways to create movement by the positioning system. This can either, two popular ways. This can either be by stepper motors or piezoelectric motors. The VersaScan system is comprised solely on piezoelectric motors to drive the three axes. These piezoelectric motors provide high resolution, long travel, and improved repeatability, all within a small footprint. This is a closed-loop positioning system. A closed-loop positioning system means that there's a feedback mechanism that moves the probe to the requested position. This is in comparison to an open-loop positioning system that requires a factory calibration and relies on that factory calibration to estimate the positioning of the probe. A closed-loop positioning system gives you that feedback and ensures the probe is in the exact position that the user has requested it. The VersaScan is installed and supported by the global network from Princeton Applied Research and Signal Recovery brands. Included with the VS base is professional on-site installation, a laptop computer to control the system, and the sales and support of the Princeton Applied Research network. There are many different types of electrochemical and materials measurements that can be made from the localized techniques. Some of those include the scanning electrochemical microscope. This is the topic for today's discussion. This is the most popular of any of the scanning electrochemical measurements, and that is based on its versatility and its high resolution. Another technique is a scanning vibrating technique or scanning vibrating probe, the localized electrochemical microscopy technique, the scanning Kelvin probe, the scanning droplet cell, and the OSP topography measurement. And we'll talk a little bit today also about how you can combine a topographic measurement with an SECM measurement to do a constant distance measuring. So let's talk a little bit about bulk techniques or traditional electrochemical techniques, and then we'll discuss the value added from an SECM. In a bulk electrochemical technique, we're going to take a metal sample and polarize it in some way. So we're going to apply a voltage to the sample, and that voltage is going to cause a current to flow. The rate at which that current flows is the rate of reactions and thus or rate of electrons and thus the rate of reactions. And we're going to convert that current flow into a reaction rate. But that assumes uniform distribution. And one thing that electrochemists can always tell you is that it's very difficult to create a homogeneous sample. Most samples are heterogeneous. They will have either local anodic and cathodic areas or local active and passive areas across the sample surface. But a potentiostat has no way to distinguish where on the sample surface is active. So that's why you must take the entire current and divide it by the entire surface area. So let's contrast that to a localized measurement. Localized measurements are made by positioning external measurement probe close to the sample surface. So these are non-contact measurements. When we say close for an SECM, we're looking for about 2 to 4 probe diameters away from the sample surface. So it's non-contact, but in an example of a 25 micron probe, you want to be between 50 and 100 microns away from the sample surface. That's going to be roughly the diameter of a human hair away from the sample surface. And so a common question is how we're going to be able to position the probe that close to the sample surface without touching it. And that's going to be one of the topics we'll cover today. It's called an approach curve. We're going to use electrochemistry to do this. But from a localized measurement, the way we can make localized measurements is we're not going to be recording the current at the sample. We're going to be recording the current flow that's happening at the tip. So we're going to apply a voltage to the sample just like we would in the bulk technique. That voltage is going to cause local current to flow at the sample, and we're going to record a voltage field at the probe. So let's look at it in the idea of spatial resolution or sampling volume comparison. So what I've done here is made two comparisons, one with the bulk measurement and one with the local measurement. And the gray rectangle represents the metal sample surface, and the blue represents basically where the samples is being measured. In a bulk technique, we're going to be measuring the entire electrolyte interface. So anything that's exposed, any part of the electrode that's exposed to the electrolyte is going to contribute to the measurement of the bulk response. So you can see essentially you're going to measure the entire electrode at one time. So let's contrast that to where a local measurement is made. If I take that same metal, I'm going to make a local measurement, but my sampling volume, thus my spatial resolution, is in a much smaller location. And then I'm going to raster. I'm going to move that probe in position in X and Y as I make current measurements across the plane above the sample surface. So let's take a second to consider then how electrochemical sensitivity is affected. Often questions of spatial resolution are asked when it comes to these kind of localized electrochemical techniques. But spatial resolution is one component of the overall resolution of the measurement. The other one is electrochemical sensitivity. If we think about taking a very large probe, the current flow at a very large probe is going to be larger because current is directly proportional to the surface area. So in a large probe, we're going to have a very large amount of current measurement. So think about as a signal to background measurement. The background is very high. It's very easy for us to measure. But in a large probe, the spatial resolution is affected, is negatively affected. If I was going to take in my chart over here of a local spot, a larger probe would provide a larger spot. So while large probes help electrochemical sensitivity by giving me more current to measure, it does hinder our spatial resolution. So let's look at it the other way. What if I take that probe and I start making a smaller and smaller and smaller diameter probe? Your spatial resolution is increased. But as that probe gets smaller and smaller and smaller, the bulk current, the current being measured to that probe becomes smaller and smaller, and that this dictates that the change in electrochemical activity at the sample surface only creates a smaller percentage change being measured by that probe. Additionally, there's more complications. As we stated earlier, we want to be about 2 to 4 probe diameters away from the sample surface. So if we're working with a probe that's only 10 microns in diameter, we need to be 20 to 40 microns away from the sample surface instead of the 50 to 100 microns that a 25 micron probe would allow us to be. So you can see that how a positioning system plays a role in how the total overall effectiveness of an SECM measurement. We're already exposing ourselves to how the probe and the probe geometry is going to affect this. And then there's the third component of the potentiostats and their ability to measure and extract small signals from large responses that is going to contribute to how well the SECM system works together to make your localized electrochemical measurement. So if I just take the word SECM and we'll break it down back to a scanning electrochemical microscope or the technique of scanning electrochemical microscopy, the imaging mechanism here is electrochemistry. What we like to say is that as a fluorescence microscope uses fluorescence as its technique to image, an optical microscope uses optics, and a thermal microscope uses temperature, this electrochemical microscope is going to look at the electrochemistry happening at your sample. And that's very exciting for us because no other technique is going to be able to give you localized electrochemical information the way you would with an SECM. So we talked about the different types of localized electrochemical measurements and localized materials measurements. All of them have different applications, all of them have different resolutions, all of them have different ways of making a measurement, but they all have the same common goal of bringing a localized character or providing spatial resolution to an electrochemical measurement. But the SECM is the most popular scanning probe technique. And why is that? It's primarily due to its versatility. I'm going to typically use two potentiostats in an SECM experiment. One potentiostat is going to be used to control the sample, and one's going to be able to use to control the probe. And so what this means is that since I'm polarizing the sample, I'm polarizing the probe, I can use these potentiostats and I can use electrochemistry and thermodynamics to dictate the reactions that are happening at the probe and at the sample. So it's an incredible versatile technique, and when we walk through a couple of different types of applications, we can use this to study anything from really any application of electrochemistry, from fundamentals to energy storage devices to corrosion to coatings to biological applications. But also we have very high spatial resolution with the SECM, the highest of any of the electrochemical techniques. This is based primarily on our ability to control the probe to sample distance and the ability to kind of control the mechanics of the system itself. So again, as we compare this to bulk techniques that are used with large-scale potentiostats and large-scale cells, in traditional measurements, the potentiostat measures an average current across the entire electrode-electrolyte interface. And so even if you have the world's best potentiostat and the world's best cell, we have no way of knowing where on the sample surface any of these responses are coming from. And that's really why we would be doing localized techniques, is to understand where on the sample surface the contribution to the total overall current is happening. And that's, just stresses the value of the SECM is the fact that samples are rarely homogeneous. If samples were homogeneous, then when we did take that integrated current and average it across the entire electrode surface, then we are presented with a uniform response. We'll know that it is correct. It's only correct if we're studying a homogeneous sample. But it was really this need to investigate localized electrochemistry, and that's due to heterogeneous samples and heterogeneous interfaces that led to the development of the SECM itself. So we discussed earlier some of the key applications. Since it's the imaging mechanisms of electrochemistry, it's all the different applications of electrochemistry itself. But some of the key ones are sensors and reaction kinetics and porous membrane studies, and then some of the other mechanistic and catalytic type applications. So we discussed the three key components of an SECM already. We have the positioning system itself. So the positioning system needs to be both high resolution and long travel to be able to look at real-world samples but still provide us the ability to get the high resolution measurements that makes SECM so attractive. But in a positioning system, we're going to need to position the probe in Z and raster it in X and Y. So we need high quality measurements in all three-dimensional axes. Then we have a probe, or often you'll see in the literature called a tip. In an SECM, this is typically a ultramicroelectrode with a specific tapered polish. We're going to talk a little bit about what that polish is and why it's important. It's crucial in doing approach curve type work. But it's typically an ultramicroelectrode, and for us with a physical electrochemistry background, it's an ultramicroelectrode disc, and it's a very well characterized type of response. Next, we're going to have we need some way to control the voltage and some way to measure the current. This is typically done with either a bipotentiostat or accomplished as a two-channel potentiostat. The difference is being a bipotentiostat is typically a single box that is designed with the ability to control two working electrodes, while a two-channel potentiostat has the flexibility to measure both the voltages and currents of two different working electrodes in the same cell, but can be split and used separately for other types of applications. And we'll say it both controls and monitors, and that's because we're going to use one to make a reaction happen. We're going to use the other one to monitor the products of the reaction. But also we've got a couple other things that are really required. We require the use of a cell. The cell is going to be some device that's going to allow us to mount and level the sample, and then it's going to also hold the electrolyte itself. Within the cell, since we're doing electrochemistry, it's typical 3-electrode type electrochemistry. We have a working electrode, but we often have two working electrodes, the sample and the probe. Then we're going to have a reference electrode and a counter electrode. And these can be common, meaning they can be shared between the probe and the sample itself, or they can be distinct, meaning that they can be separate working electrodes, reference electrodes and counter electrodes. The electrolyte. This kind of depends on the chemistry that you're trying to study and how you are designing the experiment itself. Typically, we'll use a supporting electrolyte, and so this is going to be some salt, some ionic species that are added to the liquid electrolyte in order to promote the flux in the system and reduce the migration of the species and contribution to the overall current. Then possibly we're going to require what's called a mediator salt, depending on the experiment itself. Since we're using electrochemistry to do this imaging, we need some electrochemistry happening in the sample surface. A key example of a mediator, and one we're going to walk through today, is the ferrocene-ferrous cyanide couple. There we will be doing electrochemistry on the Fe2+ to Fe3+ redox couple. At that point in time, we call that our mediator. It's the salt that's doing the electrochemistry that we are then imaging. I'm going to call out a camera as an optional accessory. This is a nice thing to have, especially in the beginning as you start doing the SECM experiments, because you want to position the probe close to the sample surface, but we don't want to contact the sample surface. And a camera helps us do this just visually as it magnifies the probe and the sample, and as the probe approaches the sample. But we also have an electrochemical way to do this called an approach curve that we're going to talk about very shortly. So here's an example. Here's a picture of a real-world SECM tip or SECM probe. They're commercially available on typically diameters that are available range between the 10 and 25 microns. These can be platinum or any other kind of noble material that is electrochemically active and kind of well characterized. So platinum is a very good choice for this. When you're discussing SECM probes and SECM tips, you're going to come across a key parameter called RG ratio. It's the ratio between the glass diameter and electrode diameter for the probe itself. So for example, if we took an RG ratio of 10, when we look at the very tip of the SECM probe, if it is a 25 micron probe, that defines the diameter of the platinum disc at the bottom of the probe. So that's a 25 micron disc at the bottom of the probe, and an RG ratio of 10 means that we have 10 times that amount of glass on either side. So it would be a 250 micron diameter glass, polished flush at the bottom, and then within that 250 micron position of glass at the bottom of the probe, 25 micron platinum disc exists in the middle of that. So often SECM probes are commercially available, but again, they are just ultra-microelectrodes. So there's many well-characterized ways to create your own ultra-microelectrode with like glass-drawn pipettes. So in an SECM system, we have potentiostats and positioning systems and probes, and we go and we make, typically we are going to be doing experiments that coordinate the movement of probe position with the movement of, with the measurement of the potentiostat. So it's that coordination and integration of the positioning system and the electrochemical control and measure that's one of the real values of an SECM system. So we have many different types of scanning experiments that we can do. These are called approach curves and area scans and line scans. So an approach curve, we're going to be moving in the negative Z direction or approaching the sample surface as we're making these measurements. In an area scan, we're going to be doing a map and the X and Y, moving in both X and Y as we measure the current. In the line scan, we're going to be moving in a single line scan in either X or Y. But with all of these scanning experiments, we're going to be looking at this as a constant voltage or constant current signal from the potentiostat. So the variable that's changing here is position. So it's a constant bias from the potentiostat, and then any changing response is attributed exclusively to the movement of the probe and the new position of the probe. You would not want to scan the probe position and scan the voltage at the same time, because then you've convoluted the response of the changing current. But we're also capable of doing dynamic signal experiments. And so these are experiments that are like a cyclophotometry or an impedance experiment, where the signal controlled by the potentiostat is much more complicated. It's either a scan or a sweep, a ramp, or an AC measurement in those two examples I just gave. But in doing those, we typically would hold the probe in a fixed position. So the parameter being changed is the voltage, not the position in those types of stationary experiments. So let's talk a little bit about different modes of operation. And we're going to come back to this a little bit in kind of a summary slide in a couple of slides. But we're going to use two electrically isolated potentiostats. And that's going to allow us to operate in either feedback mode or generation collection mode. Feedback mode typically uses a single potentiostat, and it relies on the thermodynamics of your sample to cause the balancing electrochemical reactions that are measured. It's very good for doing approach curves and measuring localized kind of conductivity changes on your sample surface. Generation collection mode has become more popular, and it's very common for sensor or kinetic type applications. And this is where we really use the two potentiostats to make the measure. This is, imagine A goes to B and B goes to another species. So either A goes to B and B goes back to A, or A goes to B and B goes to C. So we're going to use a single potentiostat in the feedback mode for doing our approach curves, and then we're going to leverage the second potentiostat then to control both the sample and the probe in a generation collection scan. Since these are two potentiostats in a single cell controlling two different working electrodes, one of them needs to be floating ground. This is, you'll hear it maybe floating ground or electrically isolated from ground or virtual earth. Otherwise, you'll have a ground loop if you try and control two working electrodes in a single cell with two earth-ground potentiostats. So the most common way to acquire data is called a constant height experiment. Constant height meaning that the Z position does not change in the course of the area map. And again, so what this does is allows us to kind of maintain a constant height or constant Z distance as we're incrementing the probe in X and Y and making these localized current measurements. So I call that modes of operation. This kind of also, either those modes of operation, either feedback mode or generation collection mode, can work in sweep or step mode. So this works, so while mode of operation of feedback and generation collection is really dictated by what you're trying to measure and the level of control you want to have in your system, the modes of data acquisitions is simply if in sweep mode the data is sampled as the probe is moving. This is typically used for faster experiments or sample exploration. And then we'll go back and do a step mode test where the probe moves to a fixed location, stops, and allows the user to oversample an average, thus reducing the noise in each of the pixels of the data map. You can define, in sweep mode, you define the sweep rate as a scan rate, as microns per second. In step mode, we're going to define a pre-delay, an acquisition rate, and a number of samples. And that, again, that oversampling and averaging reduces the noise in a single step, but it does increase the length of the experiment. So sweep mode, especially fast experiments, if you have a very large sample and we don't know where the activity is happening, sweep mode is very valuable to look at a large area in a short amount of time and identify those hot zones to come back in those areas of interest. So while most work is done as constant height, there is a very valuable variation on this called constant distance mode. And so what constant distance mode, where constant height maintained a constant z position as it increments in the x and y, what constant distance mode will allow you to do is make some topographic measurement by some means, and so basically measure the tilt or the curvature or of a sample with some complex geometry. So take an extreme example of a curved piece of sample, a curved piece of pipe. If we measure the probe with constant height, we're going to have very high response where the probe to sample distance is very small, and then as we move in z, the probe to sample distance would get larger, and that would decrease my signal response, and so that convolutes my response. It gives me the localized current is then both has contribution of a changing probe to sample distance and a changing localized electrochemical activity, and so that convolution of the response doesn't allow me to make as clear of an image or draw as clear of a conclusion from my data. So constant distance mode is becoming very popular as a way to increment the probe in z and maintain a constant distance between the sample and the probe throughout the scan. And so now we've removed the contribution of topographic complications, and now it is solely, any change in current is solely due to the changing localized character of your sample. So let's take the next step and let's talk about how we actually do these experiments, and let's show some data. Typically, I say an SECM experiment is really 5 different sub-experiments. We're going to start by doing a cyclovoltammetry experiment on the probe. We're going to then do a cyclovoltammetry experiment on the sample. Then we're going to do an approach curve over the insulator, an approach curve over the conductor to help position the probe, and then we're going to do the SECM line scan or area scan to look at the area of interest. So let's walk through exactly how we're going to do this again. So here we have the cell comprised of a 5mm gold disc. We're working with a probe that is a 25 micron platinum probe with that RG ratio of 10. Then as a supporting electrolyte, we have 100 millimolar KCl. So in the image on the left, we have a typical sigmoid shape of a response of a ultramicroelectrode in a cyclovoltammetry experiment. So my bulk, when I created the solution, my bulk ion exists as iron 3+. And so that's important to know, because if I look at the oxidizing potentials, the positive potentials on the right, we're going to have iron 3 plus exists, and there's basically no electrochemistry happening at the positive potentials because I'm already starting with iron 3 plus in the bulk state. Then as a cyclovoltammetry experiment, I'm going to take from a positive potential and I sweep negative, and then I'm going to sweep back positive, and I impose a triangular waveform of voltage onto this platinum 25 micron disc. As I sweep from the positive potential down to of almost 600 millivolts, as I sweep negative down to 200 millivolts or so, I'm going to see that I have basically no current flowing at the positive oxidizing potentials, but as I go down to negative or reducing potentials, I now have changed the iron 3 plus in my solution to iron 2 plus. So I've reduced that iron 3 plus to iron 2 plus. That causes current to flow in the system, and that current is measured at the probe here and represents about 40 nanoamps of current flow. You can see that this is a reversible reaction because as I change from a sweeping negative back to sweeping positive, I reoxidize the 2 plus back to 3 plus, and I get back to about a zero current measured at the probe. So that's the reaction couple. I'm looking at iron 3 plus to iron 2 plus, and there's some different things that I'm going to take away from this sub-experiment, if you will. Primarily, this tells me what potential I'm going to use when I go to imaging of my area maps and line scans. If I have the system as designed here for this experiment, if I were to apply a positive potential or an oxidizing potential to a solution where iron already exists in the 3 plus or oxidized state, I get no electrochemistry happening at my probe. That means no current flows, and that means my scanning electrochemical microscope is not able to make a useful measurement. However, if I move from the positive potential, if I impose a negative potential or a reducing potential on that platinum disc, I now can force the iron 3 plus to reduce to iron 2 plus. I have current flow. So the primary purpose of this experiment is to decide what potentials I'm going to use in imaging with this probe as I move on to the line scans and area maps. Also, what's nice is you're going to see this is about 41 nanoamps of current then at about 200 millivolts of voltage. So when I do my approach curves, this experiment's happened in a bulk or non-localized way, but when I do my approach curves, that's the level of current that I'm expecting to see in the bulk state before I start getting close to the sample with the probe. So now I've taken my 3-electrode electrochemistry, I've done it on my probe. Now I'm going to move over and I'm going to do 3-electrode electrochemistry on my sample. Really what I'm looking for here is that my sample is polished and clean and gives me just kind of a typical response that I expect. And you can see your duct-shaped CV gives me the response that I expect from a 5mm gold disc in a ferrocene-ferrocyanide couple. That kind of diffusion-controlled Nernstian type response. So again, let's take a step back and we're going to compare the two modes. We're going to do feedback mode, which is going to be a single potentiostat. I'm going to just monitor current at the tip. I'm going to use a redox mediator such as ferrocyanide or rubipi or some other kind of well-characterized reversible redox couple. And in this feedback mode, the sample connection may not be required. And so this is very nice, but from a sample mounting standpoint, you do not even need to make a wire connection to your sample if you're only going to be imaging in feedback mode. And this is really going to be used for approach curves, and it's going to be used for the ability to kind of measure localized conductivity and make comments on that. But a more common way of doing SECM is after the approach curve happens is to move on to generator collector mode. This is where I'm going to use 2 potentiostats. One potentiostat is going to be connected to the tip. One's going to be connected to the sample. I can run this in either way then. I can apply the signal to my sample and measure the response at my probe, or I can go the other way around and control what's happening at the probe and measure the response to the tip. And I've got some images later in the presentation that are going to make that a little more clear. In generator collector mode, you may have a redox molecule in solution, mediator in solution, or it may have what I'm referring to as an inherent mediator. So if you have the reaction A goes to B and B is electrochemically active, you can then use that itself without any external salt being applied to make that B goes to C or B goes back to A redox reaction. Generator collector mode has a little bit less spatial resolution due to the random walk or the random diffusion of ions. If you think about once you're at the sample, you're generating a response at the sample. So generically, we can say the state of the molecule ox goes to the state of the molecule red at the sample, and then flux has to happen, mass transport has to happen to get the red molecule to the probe so it can be measured. That diffusion doesn't happen linearly. Diffusion is a random process, or not random. It smooths concentration gradients, so it's not diffusing directly from the sample directly to the probe. It's expanding its concentration zone, and in doing so, it does not stay localized. And that mechanism kind of reduces its spatial resolution a little bit just because of the random walk of ions. But so in our 5-step process, we've talked a little bit about we've done a CV on the probe, and we did that primarily to determine the voltages that we're going to be using in imaging. We also did that. That also gives us an idea of the amount of current to expect from a bulk response with the probe. Then I moved on and I did a macro electrode CV or a sample CV, and that fundamentally just kind of proves this sample connection, and I have the response I'm looking for, the bulk response I'm looking for out of the probe itself. So the next 2 experiments I'm going to do are very similar. They're approach curves. In fact, it's the exact same experiment. It's just whether I'm over a insulating area or over a conducting area. So this process is the goal really is to position the probe in z-axis appropriately close to the sample. I said one of the key advantages of SECMs is high spatial resolution, and part of that is the ability to electrochemically position the probe. You'll be able to position it electrochemically better than you'll be able to position it with your eye, even with using a camera. So the process here, this walks you through, and this assumes that the bulk mediators in the ox or the oxidized state, as it is in my example here using iron 3+. If you started with a bulk mediator in the reduced state or started with ferrocene in the iron 2+, it's just kind of swap which potentials you're going to be applying. So the first step in the process, I'm going to position the tip or the probe in X and Y over a known insulating area. Then I'm going to connect the active potentiostat, or the potentiostat where I'm making the measurement, to the tip. I'm going to polarize it sufficiently to cause the reduction reaction, ox minus adding the electron going to red. Then we're going to, again, that's determined from the cyclovoltammetry experiment. Then I'm going to record the current at the tip as the probe approaches the sample in increments of z in the negative z direction. So imagine I've got a probe and it's sufficiently far that I'm getting bulk response away from the sample, and then I'm going to move the probe down in z, and typical values are maybe the radius of the probe. So if I take a 25 micron probe and I start making steps down, I want to do that in a 10 or even 5 micron steps down. And that's going to allow me to take there and image acquire the current. Then I'm going to see the response move from the bulk response to the localized response. And I'll show you some examples of what that looks like and some rules of thumb or practical advice on when we can stop these. Then I'm going to take this probe, I'm going to move it, it's going to return back to the bulk distance. Then I'm going to reposition it over X and Y in our conductive area of the sample. I'm going to run that same experiment again. I'm going to then overlay those 2 approach curves, and that's going to tell me where to position the probe for my line scans or area scans, and it's also going to give me an idea of what kind of responses I can expect from here. So I told you we're going to move from the bulk response to the local response. Now, what does that look like, and what is the fundamental mechanism that's occurring that's causing it to occur? So if I look at my image here in the top left, the bulk response, I've drawn out what the SECM probe is going to look like, and it's coming down the shaft of the electrode. It's coming down, and then it's going to start doing this taper in, and then I'm going to get down to that kind of flush polished glass platinum at the tip, interfaces at the tip. And so I'm going to take the potentiostat, and I'm going to polarize it to the level dictated by the cyclovoltammetram. So in my specific example, I've got the iron 3+, and I'm going to polarize the tip to a negative 250 millivolts. And so in a bulk response, that's causing the ox, or the iron 3+, when it touches the platinum, it donates the electron and becomes the reduced species. So that's my iron 3+ to iron 2+ reaction. That causes the current to flow at the 40 nanoamp level for this response. Here I have a constant voltage being applied. Then I'm going to increment it in Z. I'm going to approach an insulating surface. Once I get very close to the sample surface, you can almost consider it as a thin layer cell, or as a small electrochemical cell. Is no longer the entire volume of solution. It has now reduced itself to just the volume up under the, or in contact with the platinum wire. So you can see in the bulk response, we're constantly refreshing the concentration of ox at the platinum wire, and so my current is a high steady state level. But as I move down close to the sample surface, I've now consumed the local concentration of ox, and the bulk concentration of ox no longer has the capability to reach the platinum wire. So you can see now why the specific taper and the dimensions of the probe are very important. If I had a very thin piece of glass, or the RG ratio is a very small number, that would still allow the ox target ion to reach the platinum wire. You know, take the even more extreme example of no glass at all. We would still have access of the ox to the platinum wire, and our current would still be at the localized level. So we seal that in glass. That allows us to restrict access of ox to the platinum wire over insulating surface. So that is what is happening from the probe standpoint in the system. So what's happening from the response? How do I see that? How's that measured by the potentiostat? Over here on the right graph, I've got current on the y-axis, and I've got the Z on the x-axis. And it's important to know the way I have it graphed here, the Z approaching the sample is going from left to right. So at the samples on the far right, at the bulk is at the far left. And you see that you have a fairly steady bulk response happening as we approach the sample surface. Once we get to the sample surface, the localized response starts happening. And what do we see that as? We see that manifest itself as a quick and dramatic change in the measured current at the probe. So while the potential has not changed at the probe, the current is changing. Now, the current changed in the cyclovoltammetry experiment because I was sweeping and I was changing the voltage and keeping the probe at a fixed location. The current's changing here, but for a completely different reason. The probe is held at a constant voltage, but because the position is changing and the mechanics of the cell are different then, and I'm no longer allowing access of ox to that platinum wire without the ion at the platinum wire, no electrochemistry is happening. No electrochemistry happens, the current turns off. And we see that bulk response then come here in the bulk state and then go down dramatically. Then we're going to stop that response before we get to zero. Theoretically, you would get to zero at the sample surface, but we want to stop that a little earlier just to account for the change in any kind of topography effects or any kind of tilt in the probe or the sample itself. So that's interesting enough, and it's very easy, or it's not very easy, but once we understand the electrochemical mechanisms happening in your cell, we can see why current approaches zero or kind of turns off as the probe gets very close to the insulator, and our electrochemical cell is reduced to a small volume that is where all the ox has been consumed. So then I'm going to return, the probe returns to the bulk distance, and I'm going to move it over what's in the conducting area, or move it over the gold part of the sample. Then I just repeat the exact same experiment. Basically, I just run the exact same experiment. I'm applying the same voltage. I'm incrementing in Z in the exact same way. But what is happening when I get very close to the conductor? What's happening is precisely kind of depends on whether I'm in feedback mode or generator collector mode, but in this example I'm talking about feedback mode because we're going to do more generator collector work in a little bit. But again, the probe can only see, again, if we think about this as a microscope, an electrochemical microscope, it can only see electrochemical reactions that are happening at the platinum wire. That platinum wire is only exposed to a very small subsegment of the larger volume of the cell. So the bulk state is exactly the same. I have negative 250 millivolts applied to the platinum wire. We have ox, the iron 3 plus, going to iron 2 plus, the reduced species, and I get about the same amount of current flowing out here. It's very far away from the sample. But as I approach the sample, the mechanism, the local response, and the mechanisms that happen due to the local response are very different. What's going to happen here is as the electrochemical reaction turns ox to red, or in my specific example, the iron 3 plus to iron 2 plus, I now have a high concentration of iron 2 plus at the probe, or at the small subsegment of volume below the probe. So now I've got 3 plus has gone to 2 plus at the reduced species. Now I have a very high concentration of 2 plus at the conductor, at the gold, at that level. So what is the Nernst equation and thermodynamics of that gold going to do? Even if it's non-polarized, in feedback mode, there's no connection to the gold at all. In this non-polarized state, what's going to happen is there's going to be a balancing reaction on the gold substrate in order to maintain that thermodynamic equilibrium, thermodynamic level. I'm going to have a balancing reaction that's going to cause the flow of electrons to happen. So a balancing redox reaction happens to account for the high concentration of reduced species at the probe. Somewhere else in that gold, I'm going to have a balancing reaction happen. And so that's not seen by the probe because it's outside its sphere of seeing. It's outside its ability to make that measure. So somewhere else in the gold surface, I have a balancing reaction happening, and then this allows the high concentration of the reduced species to once again become the oxidized species at the probe. This, again, I'm using a specific example of a reversible reaction, and this is A goes to B and B goes back to A in a generic interpretation of the electrochemistry. So we have ox goes to red, red goes back to ox. What does that do then? It increases the local concentration of ox at the probe. That causes another reaction to happen from ox to red. That causes a self-propagating kind of cycling or a positive feedback response. And so the closer I get to the conductor and the probe, the more this recycling happens, and the more current happens. So the higher the localized concentration of ox happens, so the higher current is seen by the probe. And so we can see this in the response. Again, I've got current on the y-axis, and I've got approaching sample from left to right on the z-axis, on the x-axis of the graph. I'm going to have about the same bulk response happening on the current, but then when I get very close to the sample surface, this localized response is seen in the opposite direction. So it's a similar shape, but instead of reducing the current, the current actually increases. And from a fundamental electrochemical point of view, I find that very interesting because this happens even though the conductor is not polarized at all. So this allows me, I'm able to see and measure the conductivity on the sample surface without touching the sample surface. So again, that's very interesting from a fundamental study of electrochemistry. But it has a practical use in an SECM experiment as well. Because now I know where to position my probe in Z to make these measurements. If I go back then, I can overlay these two scans, the approach curve insulator and the approach curve over the conductor. Let's overlay these responses. We can see both are giving similar type current in the bulk response, and then as the probe increments down and down in Z closer to the sample surface, both of them provide a localized response. But the localized response over the insulator makes the current go down at the tip. The localized response over the conductor makes the current go up at the tip. And so once I overlay these, I know now where to position the probe, and we can even see and kind of predict the impact of probe to sample distance on electrochemical resolution of an SECM experiment. Because the Z position shown x-axis on this graph goes to 0 to about 1,500 microns. So if I was at the 0 position, so 1,500 microns above the ideal position here, if I took a scan, an area map with the probe at that level and the plane 1,500 microns above the sample surface, what would I see? Well, the response over the insulator and the response over the conductor are the same. So since those electrochemical responses are the same, and since my microscope is based on electrochemistry, I have no way to distinguish where is high activity and where is low activity. But as I start bringing the probe closer and closer to the sample surface, and you can see this grid line right here is 1,400 and then microns, I start seeing if I was to position it at 1,400 microns closer to the sample surface, we can see that I would be getting approximately a 48 nanoamp response out of a insulator and about a 51, 52 nanoamp response out of the conductor. So while that's appreciable and what's statistically significant, it's only about a 4% difference from the bulk of these two responses. So that gives me a very small difference between a highly conductive and a highly insulating response. But you can see this almost asymptotic type relationship as we move into the sampling zones closer to the 1,500 micron response. You can see that you're going to expect in the 30-something nanoamp response over the insulator and the 70-something nanoamp response over the conductor, which is more than a factor of 2 increase in the current response over the insulator and conductor. So you can see, think all the way back as we talked about spatial resolution and the drive is to go to lower and lower probe diameter, smaller and smaller probe diameters. What that does, it would be to force that bulk response to a lower and lower level. That complicates or decreases the ability to see and to image at over insulators and conductors. The ratio of the insulator conductor to background becomes smaller, and so that's seen as the effective decrease in electrochemical sensitivity. So often we're asked the question, how do I know what size probe to use? And it's really you want to use the largest probe diameter. It's still going to give you sufficient spatial resolution to see the images that you're looking for, or to image the sites that you're looking for. So once we have taken this probe, I know that I need to move 1,500 microns closer to the sample surface, and with software you can easily position the probe or tell the probe to go to 1,500 microns closer to than its relative Z position is right now. I'm going to move that down, and then this is where I've now accomplished 4 of my 5 sub-experiments of the SECM. We've done the CV of the probe, we've done the CV of the gold, we've done the approach curve of the insulator, we've done the approach curve over the conductor, and now that I am in a position to measure insulators, conductor, and kind of run this in an area map experiment. And so this is a very small area map over just showing the gold transition, where the gold is on the far left-hand side and goes to the the gold's on the far right-hand side because it's lower current, and then we move into the gold response, and you see the response showing up as a purple response, the higher current response there over the gold. The right-hand side is these same data just represented in a 3-dimensional rotational type format. So that's a lot of feedback mode. We talked about approach curves and how we do the experiments, and showed some real data taken on gold sample and a platinum probe with a ferrocene-ferrocyanide redox couple. So let's talk a little bit about substrate generation, tip collection, and then the tip generation substrate collection experiments. You'll see this abbreviated SGTC often in the literature. So here we're going to use both potentiostats. One potentiostat is going to be connected to the conductor, the sample. One potentiostat is going to be connected to the tip. And I'm going to be able to polarize them independently and kind of dictate what's going to happen. And so if we can think about this as really, I am imaging concentration gradients at this point. Don't think about this as imaging of the sites or imaging the sample. In generator collector mode, what I'm really imaging is the electrochemical field or concentration gradient created by the site. And so if I take a generator sample, again, I'm going to use this whole disk. I've made my solution to be solely 3+ iron in the beginning, so ferrocyanide. Now I've applied a reducing potential to that gold sample surface. And so what that does then is it causes iron 3+ to go to iron 2+, all on the sample surface. Since iron 2+ does not exist in the solution, in the bulk state of the solution, diffusion is going to take control, and that iron 2+ then is going to diffuse away from the sample surface. So now that iron 2+ is starting to propagate, that concentration gradient is starting to propagate out into solution. And you can see this kind of drawn as a concentration gradient on the right-hand side, concentration distance from the electrode X, but here it's Z as it's fundamentally propagating upwards and Z and closer to the sample, to the tip. What I can do on the tip then is add or polarize the tip to a certain potential as well. But I don't want to polarize it to the same potentials I've polarized the sample. So the sample I'm using to generate 2+ from 3+. Think about that, and then the terminology and nomenclature becomes clear. I am using the sample to generate another electrochemical species, and so, or another electrochemical state. And so I'm then reducing 3+ iron to 2+ in solution. That's going to propagate up, and I'm going to eventually that's going to reach the tip. I put a oxidizing potential plus 650 millivolts on the tip. That's going to cause, remember, no electrochemistry happens on the 3+ iron at plus 650 millivolts. But if the iron 2+ comes in contact with a platinum wire that's been polarized to 650 millivolts, I now cause the oxidation of iron 2+ back to iron 3+ in current flows. So we can see how the terminology comes from generator and collector, because I'm using the sample to generate iron 2+, and I'm using the collector, the probe, to collect the iron 2+ generated at the sample. that's going to let me do then, of course, is look and see the more active sites, more active positions on the sample are going to generate more iron 2+, and that's going to be seen at the probe by an increase of current at the probe, because the probe's where is measuring the iron 2+ to iron 3+ transition. For users that have experience with rotating ring disk, RRDE work, this is very common type of a thought process, as you're using the disk to generate one species and you're using a ring to collect that species. But this gives you spatial resolution, so this is the next kind of step in a RRDE type kind of a thought process, I guess. The current at the sample is going to be very large because current is proportional to surface area. If you had looked back at the current magnitudes measured during the cyclical voltammetry experiments, where I'm measuring tens of nanoamps on my microelectrode, I'm measuring hundreds of microamps on my macroelectrode or my gold, so about a 10,000 factor higher current. You're going to see very low current where there's no iron 2+ at the probe, because my background's virtually zero. And then as I raster that probe across, and when that probe comes into contact with the diffusion field or the concentration gradient of 2+ from that sample surface, I'm going to see that as current measured at the probe. And so as my tip to substrate distance decreases, then my current increases, because that probe stays more localized and that field becomes larger, and the current is really, it's going to be proportional to the slope of that field. I can run the exact same experiment in a different way in tip generation substrate collection. The connections are exactly the same. All that happens is you redefine which one's generating the iron 2+ and which one's collecting the iron 2+. So in this diagram, we have the probe that would be receiving the negative potential to reduce the 3+ to 2+. Now I have a concentration gradient that's propagating itself away from the probe that's going to be collected at the collector. So I'm going to see a kind of constant current at the tip, and I'm going to see a change in current then at the collector as the tip moves from an insulating into a conducting region. The bulk response is going to be much lower because there's much less 2+ being generated in this scenario, because the surface area of the platinum electrode is much, much smaller than the surface area of the gold. So you can see how we can go back to the substrate generation tip collection slide, and you can see here we're using gold and platinum, so I need this mediator to exist in the iron 3+ to iron 2+ state and back to 3+. But commonly in a sensor or in a catalyst type experiment, or you may be able to design the experiment in such a way that a electrochemically reactive species is created by the initial reaction at the collector. And so to put that in practical terms, what I mean by that is if you have oxygen being created as a byproduct of your electrochemical reaction at your sample, then we can use the probe and set it up to study the oxygen reduction reaction. Wherever you have the oxygen reduction reaction, that means that you have your reaction of interest happening at the probe. The current dictates the relative rate of reaction, charge is the quantity measured. You can also see this as a porous, as an example when studying porous membranes. If you were to put a membrane between the tip and the collector itself, then as the species have to have restricted diffusion, they have to diffuse through the pores, and then you can take the probe and raster in the plane above the membrane, and you can see where the active ion or the target ion is coming through the pores. And again, you see with electrochemistry, so you see by measuring the current at the probe. So topographic effects and complications. So approach curves, if we think about this, you saw the asymptotic relationship of current to probe to sample distance, and not just saw it, we quantified that in an approach curve. And so you can see the probe to sample distance really impacts, because we want to operate in that high differential area, so that probe to sample distance really impacts the measurement in that region. So that tells you how important it is to maintain a constant probe to sample distance as we're doing these experiments. Now, if your sample is polished flat and not tilted, the constant height experiment can make these measurements for you. But if you have more complex geometries with curved samples or sensors that may have some topographic character to them, this is where we're going to need some way to measure constant distance, save that as almost like a background file, and increment that probe in Z as it goes across the plane above the sample surface. So again, we talk often about some sample resolution, some resolution considerations, as that is one of the most common questions when we discuss the SECM, is what's the resolution of your SECM? Really, the resolution of the SECM it's not the hardware itself, it's the resolution of the system and the resolution of the experiment, and that's as dictated by the chemistry as it is different components of the system. But let's walk through a couple key parameters of resolution considerations for an SECM experiment. First, we're going to have both the accuracy and the resolution of the current measurement. So this is done by the potentiostat. So not only what currents can the potentiostat resolve, but for quantitative work, we need to know how accurately it can measure these low currents. Then there's the accuracy and resolution of the positioning system. Again, it's how well can I report back and how well can I position the probe in a known position, and once it moves away from that position, how well can I control it when it comes back. So knowing the resolution and accuracy and repeatability of a positioning system is a key parameter in the ability of an SECM system to make the measurement. Probe diameter, and this is the one that does get clear to, is clear to many. As we reduce the probe diameter, you will increase your spatial resolution. But as we spoke about a couple times in this presentation, it's not all about probe diameter, because that decreasing probe diameter decreases the bulk current that flows at the sample, and it flows at the tip. This makes the ability for accuracy and resolution of the current measurement even more important, but then it also makes that ratio of the current that flows over the insulator and current that flows over the conductor, it just decreases that. So it's going to decrease that kind of electrochemical sensitivity. Another thing to point at, something is going to limit or something is going to dictate the spatial resolution or the resolution of the measurement. If you were looking to improve the resolution of your measurement, what you're going to need to do is take all these kind of considerations and work to improve the one that is limiting your spatial resolution. So for example, if you are limited by your topographic effects, you can use the world's smallest probe diameter, and it does not increase the spatial resolution or electrochemical sensitivity of the response at all. You need to improve the one that is limiting the resolution of your system and of your specific measurement. So let's walk through this a little bit further. Then topographic effects we've touched on a couple of times today, and that is if you have tilt or complex geometry of your sample surface, it's going to convolute, or it's going to have two different parameters influencing the current measured at the probe. Then we're going to look down and we're going to see probe to sample distance as an impact. This is quantified by our approach curves. The closer you get to the sample surface, the better that ratio of current over the conductor and current over the insulator becomes a higher value. So really that electrochemical sensitivity becomes higher. But controlling that relates, there's an interplay to the topographic measurements above, and it's also an interplay with the probe to sample for the probe diameter, because as that diameter decreases in the probe, we need that probe to sample distance to get smaller, and then you have even more of a possibility of having topographic effects. So these all go hand in hand with each other, and a lot of thought has to happen to determine which one needs to be improved to improve the total resolution and measure and accuracy of the experiment itself. And then, as we talked about, we're eventually going to be limited by the random walk of ionic diffusion as diffusion happens, and diffusion carries the products of the reaction at the sample to our electrochemical probe. And so we gave a talk in a conference in Germany specifically on the fundamentals of the SECM and each of those fundamentals and how each of those impact the resolution. You can see that here, and it's available from the Principad Research website as well. So throughout this presentation, I've tried to give some examples of how SECM would be used in different types of applications. Here are a list of several different applications where there are different publications in the literature. Just imaging, ultramicroelectrode shape characterization. So once you've made these ultramicroelectrodes, you can use them to characterize themselves almost as in SECM work. Heterogeneous kinetics. This is what we're talking about, kind of the corrosion mechanisms. Homogeneous kinetics. Biological systems. A lot of the field of bioelectrochemistry is a rapidly growing field, and there are some certain applications of SECM to biological applications that we talk about over here on the right specifically. Liquid-liquid interfaces, membranes. We talked about how by putting a membrane between a high concentration of analyte and the low concentration of analyte, you force diffusion through the porous membrane, and then we can measure the pores and the rate of diffusion then through the pores by scanning the probe across in the low concentration section of the electrochemical cell. Surface reactions, semiconductor, small volume electrochemistry, and then we can also do fabrication with SECM. You can either design it to plate in a localized way onto your sample, or to strip. And so this would be a fabrication. If you wanted to add material, you could set it up where it would plate onto the material. If you wanted to decrease or create valleys, you could just reverse the potentials and cause stripping to happen at the sample. But sensors, fuel cells, these all are applications that benefit from localized electrochemistry. So I gave some examples of how generator collector mode are used, and you can see this and how valuable it is if you can design the experiment in a way where some other electrochemical species is generated from the primary reaction of interest. We can study that without using a mediator. And so if you use molecular oxygen, you can use a oxygen reduction reaction to study that. And then there are several examples of literature in imaging a live cell in doing so. And where are you going to know where the live cell is? Well, when the molecular oxygen concentration decreases, that's going to be where the live cell is, because that cell is taking in oxygen and putting out carbon dioxide. Think about the same experiment but in a different application to study photosynthesis, where the plant is going to be taking in the oxygen where the plant is going to be producing the oxygen from the carbon dioxide. And so what's going to happen there is over the live cell would be imaged by a decrease in oxygen content shown in your SECM as a decrease in current, and then on the oxygen regeneration side for photosynthesis, you're going to see that at the tip by an increase in current, and that's because of the increase in oxygen that will come from a photosynthetic reaction. So in addition to this web seminar, we have a couple different pieces in our SECM library. We have the full poster on the fundamentals of scanning electrochemical microscope or microscopy and the impact on those resolutions. This specific presentation on an SECM, and then a small book chapter on the SECM that was published entitled Electrochemistry and the Fundamentals of Solved Problems. So that concludes today's installment of the educational series presented by Princeton Applied Research entitled The Scanning Electrochemical Microscope. Thank you for your attention, and please feel free to contact us with any questions as relates to this or any products that we offer. Have a good day.